All right, so hello, I am Kate Sheehan at Frostburg State University, and today I'm going to be talking about the use of stable isotopes and parasite community assemblages and our ability to use those to differentiate among waterbird groups. This is a highly collaborative effort that has progressed throughout several years. We're really wanting to combine the use of biogeochemistry and more biological, ecological indicators to understand how organisms are using their habitats. So I'll start off by talking about the fact that we as humans like to modify things around us. We tend to change our environment to make it more comfortable for us. And in doing so, a lot of times we will change the landscape. And consequently, wildlife is going to respond to that by modifying their distributions or how they're using the resulting habitat. So these wildlife are responding to changes in available resources. And some of those resources would include things like breeding sites or nesting supplies, things like that. Migratory birds, as they're moving over a landscape, are going to cue in on specific types of habitat that they might use as stopover. And then, of course, for energetic needs, they're going to be eating and drinking different things on this landscape. So one of the ways that we've changed landscapes is by modifying standing water bodies. And we've created them in a lot of instances or potentially diverted waters. But we do this in order to improve our ability to recreate. We've created reservoirs that we use for municipal water and hydroelectric, things like that. But another way that we have modified standing water is the creation of aquaculture facilities. And this is especially true in the Southeast and South Central United States. And we farm quite a few different types of organisms. Some of those we consume directly, and some of those are used more indirectly. And baitfish aquaculture is an example of kind of a diverse group of organisms that we farm, but we're not necessarily consuming them outright. Instead, we're using them to supply anglers who are then using them in recreation. So here we have some examples of baitfish that are farmed in the southeastern United States. And these are 4 species that you're going to be seeing data for today, or at least these are the culprits that we're trying to kind of identify whether or not particular birds are foraging on their ponds. In addition to baitfish, as I mentioned, we also farm food for ourselves in terms of fish. And one of those most commonly farmed species in the Southeast is catfish. So we take those into consideration as well. However, we also know that catfish and some of these other species are naturally found within the environment in the floodplains and the river systems, things like that. And I'll be talking about that a little bit more later. So wildlife are queuing in on these aquaculture facilities, and water birds especially will use the aquaculture ponds for foraging and stopover habitat, loafing, things like that. And they can be a problem. And when they're consuming fishes, there is a potential loss of fish biomass and the revenue that goes along with that. In addition, a lot of water birds are definitive hosts for parasites, and when they visit these ponds, they can spread these parasites and pathogens that can get the fish sick and cause additional revenue loss. Defecation by these birds in the ponds redirects nutrients and can also impact water quality. But it isn't necessarily all bad. We understand ecologically that predators foraging on prey populations in many cases, remove weaker or sick individuals from those prey populations, and that's likely happening within these fish ponds as well. And then not all of the birds that are visiting the aquaculture facilities are consuming the fish themselves. Some of them are going to be eating invertebrates, seeds, things like that in the ponds, and not necessarily removing fish directly. In that way, they could be removing other intermediate hosts and reducing disease within the fish populations as well. So it's not necessarily all bad. However, for the most part, when fish farmers see birds on their ponds, they tend to be pretty alarmed about it. So something that we were trying to learn about was whether or not we could tell which birds were using which types of bait fish ponds. And I had showed 4 different types of bait fish on a previous slide, right? My co-authors were really interested in trying to differentiate among the birds using biogeochemistry. So in this case, using stable isotopes to differentiate among individuals. And this is something that we have a pretty good understanding of, right? So there are a few different stable isotopes that we can use to understand the trophic ecology of an organism. One of those is going to be carbon. So, and when we're looking at stable isotope chemistry, we're looking at the ratios of typically heavy isotopes to the more typical standard isotope of a particular element. So for carbon, it's carbon 13. And here, when we're dealing with carbon, we can we get an understanding of what the base of the food web is going to be. Okay, so if we have C3 plants, right, which would be something like soy, and soy is used in some fish feeds, we should be able to differentiate fish that are being fed soy-based products as opposed to corn-based products, which would be C4 plants. Using carbon, we get an understanding of the base of that particular food web as opposed to algal based. So these maybe would be more natural, not necessarily C3 or C4. That would be somewhere in the middle. We can also use nitrogen, and nitrogen we understand fractionates through trophic interactions. So we see an accumulation of nitrogen 15 compared to nitrogen 14 as you move from prey to predator. So we have higher concentrations in our predators than we do for organisms at the base of the food web. So using kind of a combination of those things, we've been able to, split up in different food webs, different types of organisms. We also used sulfur isotopes, or tested for sulfur isotopes, understanding that different water bodies can have different sulfur signatures in there. So that might also help differentiate things just a little bit more. But really it's using the combination, all 3 of these stable isotopes, to basically build a model or an index that could be used to predict where a particular bird has been foraging based on its biogeochemical signature, right? And that helps us to understand how they're using these fish ponds. My component of the research as a parasite ecologist was to add yet another layer onto this using trophic tags, which are the parasites themselves. And we're going to add that into the model or into the index to really try and further differentiate things. If we know that a bird has been foraging on a pond of golden shiners, it's going to have the parasites from those golden shiners in it, whereas a different bird does foraging on green ear sunfish are going to have parasites associated with that fish instead. So we should also see differences in the parasite communities if the birds are foraging on different ponds. So a little bit of background on this. The project itself started out as master's projects for 2 students who are through the program now that went through Mississippi State University. They were collecting birds from Arkansas and from Mississippi, and you can see kind of these areas are pretty close to the Mississippi River floodplain here. The bird species that we're working with are lesser scaup and double-crested cormorants. And the ducks were foraging on the baitfish ponds, whereas the cormorants were not necessarily found on the ponds themselves. So we expected that they were foraging nearby on more natural food sources, which is mostly going to be catfish, yellow perch, things like that. But we considered them as eating catfish. Okay. So the cormorants themselves are kind of almost a control because they weren't necessarily on the baitfish ponds. And we also know, oops, sorry, here's a picture of the cormorants. We also know that these two different types of birds forage really, really differently, not only based on where they're foraging, but what they're eating. So scaup are going to be omnivorous. They're going to eat some fish here and there, but they're also eating a lot of invertebrates, and they're going to be eating vegetation and seeds too. Whereas the cormorants are just eating fish. So trophically, they're very dissimilar to one another. And then behaviorally, in terms of where they're foraging, we also knew that they were pretty different from one another. So the question is, can we predict the groups of birds that are foraging on different baitfish ponds and the cormorants that weren't using the ponds? And so how do we do this? Well, I use discriminant analysis. And what this does is it builds a model based on the metrics that you enter into that. And then it takes your data back out and makes a prediction, right? It classifies, in this case, each bird as to what type of pond it thinks that that bird is going to be foraging on, either one of the 4 baitfish, or we called cormorants catfish eaters. So based on this, we know that there were 5 types of fish groups that were being consumed. We said the cormorants were eating catfish, the scaup are eating 4 different types of farmed baitfish. When both birds are included, that's 5. So the null model for this, so if we randomly assigned a bird to a pond, we would expect to be right only about 20% of the time. So we'd expect that the null model would misclassify a bird 80% of the time. Okay. Now I'm also going to show you where I ran this without the cormorants included. So in that case, we had 4 different types of fish. So we'd expect misclassification 75% of the time, and getting it right 25% of the time. So those are kind of the numbers that we want to keep in mind. So when we run a discriminant analysis, we get that kind of misclassification number. We also get an ordination plot where we have points, and each point represents a bird here, where the closer the points are, the more similar the metrics were that were put in. So if we're including stable isotopes or parasites or whatever we put in, the closer the points are, the more similar those 2 birds are. And then we have these circles, these 95% confidence ellipses, where we expect 95% of that particular, in this case, it's the fish type, or the birds that ate a type of fish, to be found around. So this blue ellipse here on the right is the catfish ellipse, and we find most of the cormorants have been placed in this general vicinity. Okay, so here are actually the results from one of these discriminant analysis models. And this is the one that includes all 3 of the isotopes. So what's been entered into this model is the isotopic data for carbon, nitrogen, and sulfur. And in terms of the accuracy of the model, it correctly classified birds 70% of the time. So it only had a 30% misclassification rate, and that is way better than the null, which is 80%, right? So we're really outperforming the null model here. It predicted all of the green ear sunfish correctly, but everything else had, some things that they're a little bit misplaced and misclassified. I was also interested in differences between the hosts themselves, because in terms of their demography, males and females could be feeding differently from one another. And we did see pretty good differences in body size as it related to some of the parasites, which I'll get into in a minute. But I wanted to add bird weight into the model as well. It improved accuracy just a little bit by 1%, but not by much. But it did allow us to achieve perfect separation between the ducks and the cormorants. Okay, so let's talk about the parasites now. So as we went through the gut contents for these birds, we found lots and lots of parasites. And this is the contingency table, where on the x-axis, we have a list of all of the different types of parasites that we were able to find between the ducks and the cormorants. And then we have 5 different kind of color blocks here that represent the different types of fish ponds where those parasites were found respectively. Okay. Now, if I were to enter in all like 20 of these parasites into the model, it would make the model incredibly difficult to interpret. So I wanted to squash down the parasite components that we were adding into the model into as few variables as possible, but I didn't want to lose all the information about the parasites. So what I did was I went through and found parasites that were particularly telling about a particular type of bird or a particular type of fish pond. And that left us with 8 different parasite types. But I wanted to squash it down even more. So I ran a principal components analysis using these 8 parasites. And that dropped everything down by half. So we went from 8 species to 4 axes, explaining 80% cumulative variance here. In reality, we only needed the first 2 principal components, the first 2 PCs included in our models in order to achieve some serious differences. So when I add parasites into the discriminant analysis, oh, sorry, before I talk about that, I want to talk about the different ways that I dealt with the data here. So I ran these models in a couple of different ways because the way that parasites impact their hosts, the way that they're represented within their hosts, it can differ depending on what your questions are and how you're treating your data. So one way the parasite information can be represented is in numeric abundance, or just counts, how many are there, and that can be really important. But it doesn't necessarily take into account, or it doesn't necessarily represent the impact that those parasites might be having on their host. In order to do that, we might take maybe their body size, the energetic restrictions that they're putting on their hosts into account. So using parasite body size combined with their abundance, we can figure out the volume of parasites that is occurring within a host, and then convert that into the energetic demand that a host is encountering based on its parasite community. And here I have an image of a microphalid, a meritrimid, trematode, and just so you understand the size of this organism, this scale bar here is 100 microns across, so 1/10 of a millimeter. So this is a parasite that we find in cormorants. This is a Phyllochasmus rather, that we find in scaup. Here, this scale bar is 1 millimeter. So if we were to, kind of compare these guys on a closer scale to one another, right, the meritrimids are much, much smaller, and you're going to have a lot of these accumulating in order to have the same kind of energetic demand that you would on a host. And then we take things like tapeworms into account, and this is a shrunken down version of a tapeworm. In reality, that's how big it would be compared to this other trematode here. Some of this can really add up. So I ran these models using not only count data, but also volume. I also ran non-parametric and presence and absence for all these models. So in all, it ended up being like 60 models that got run. These are kind of the take homes though. So the full model includes both host species. We have cormorants and scaup in here. And this one includes isotopes, the weight of the host, and the first two principal components for the parasites. And so we went from having a 71% accuracy now to 79% accuracy. So we had another pretty good jump. Only 21% of the birds are now being misclassified. So this is a pretty substantial and a good step. I don't know that we're going to get better performance without having a much more complicated model built to try and explain these things. But, you know, as I had mentioned before, cormorants, they're not necessarily, even though people don't like them, they're not necessarily the things that are sitting on these baitfish ponds. So I wanted to split and rerun all of these models again, just with the scaup, and this is what we find here. This model, the best model, wasn't quite as good as with the two species of hosts included, but that's to be expected because the other one split the cormorants off and increases success substantially. Here we still have 65% accuracy, so only 35% misclassification, whereas the null model for this again was expected to be 75% misclassification. So this is definitely still outperforming the null model. And just for comparison purposes, this is kind of just wrap up the results here. You can see two different types of models. One of them has both birds in it. Okay, and the model itself, we have either carbon isotopes, adding nitrogen isotope plus the sulfur isotope, host weight plus parasites. And we have the correct classification here. With the scaup only, we get up to 65%. However, it's a significant outperformance of the null. So these are pretty good predictive models when it comes down to it. It's way better than what we would predict just by chance. So it seems that the isotopes themselves are definitely good at helping us to predict where particular birds are foraging. They provide good information in this instance where we're dealing with freshwater farmed fishes. Okay. In terms of demographic differences, didn't necessarily improve the model a whole lot, but brought it up by 1%. However, inclusion of the parasites really did enhance the models just a little bit more. And this makes sense because we understand that the parasites are there because of what the birds themselves are eating. And if they're eating a specific type of fish, they're going to have parasites that are associated with it. So by combining the biogeochemical and the parasite related trophic ecology estimates, we seem to be getting even more and more accurate models so that we can understand how wildlife are reacting to the changing landscapes that we as humans continue to modify. So I am going to leave it there at this point and thank you for your attention. And I'm happy to talk with you all at the panel, or if you have additional questions, my email is on the screen here. Thanks a lot.